Bismillahirrahmanirrahim. In this video, the process of thrombosis in the healing is described. Following are the references for this video. A thrombus or clot is formed at the site of the injury, and the superficial part or exposed part of this that is dried up is called scab or ishkar. It forms a barrier on the wounded skin to the invading organism, and it also prevents the loss of plasma and tissue fluid. Hence, it seals the site of the injury. It is composed of plasma fibrin, fibronectin, that is adhesive protein, and various contracting platelets that are actually the source of initial burst of the growth factor. There is injury that leads to the fibrin, presence of the fibrin, and this fibrin is bound to the fibronectin, the adhesive protein. There is progressive cross-linking by factor 13, that is a transglutaminase, and it forms glutamyl lysine cross-links between proteins that form the clot and extracellular protein. This cross-linking also aids in clot retraction. Then tissue transglutaminase, these transglutaminase or transglutaminase 2 fosters cell adhesion, cell migration, and organization of the wound extracellular matrix. They actually are responsible for interlinking matrix protein, fibrinogen, fibronectin, collagen, and vitronectin. They provide local tensile strength and also maintain closure during the formation of the new extracellular matrix. If there is excess transglutaminase presence, there is undue scarring of the site, and if this factor 13 is deficient, there is poor wound healing and bleeding. So thrombus seals the area of the wound and forms a protective barrier that prevent the infection and also prevent the loss of the plasma. The main component are fibrin, fibronectin, activated or contracted platelet, and their growth factor that are stored within. And they bind fibrin. There is binding of the fibrin to the fibronectin, and this is achieved by cross-linking by the factor 13, and there is a stabilization of the clot via glutamyl lysine cross-links that aid clot retraction. Then there is role of transglutaminase 2 also, the tissue transglutaminases that enhance the adhesion of the cell, migration of the cell, reorganization of the matrix protein, and excessive amount of these tissue transglutaminase 2, they can cause scarring. If there is deficiency of factor 13, there will be poor wound healing and bleeding due to lack of stable clot formation. Then mononuclear cell, connective tissue, and vascular cell invade the internal matrix and transform it into granulation tissue. And ishkar is actually, scab or ishkar is actually a temporary repository for dead neutrophil and bacteria until the healing progresses. Finally, it detaches as the migrating epidermis position. They come together and they partition the scab or ishkar from the newly forming epidermis or epithelial tissue, and new tissue replaces the clot. This illustration is a detailed diagram of wound healing at the stage of inflammation that is transitioning into tissue repair. Specifically, this is highlighting the role of the thrombus and growth factor signaling. The thrombus that is present over here in the middle, it is composed of fibrin meshwork, platelet plug, neutrophils, and macrophages. The primary function of this thrombus that when dries on the surface of the skin is termed as scar or scab. Its primary function is to act as a temporary barrier to prevent infection and initiate the repair process. The inflammatory cells that are uh, active in this stage, in this process, are neutrophils that are the early responders, and they also phagocytose the debris and bacteria. While the macrophages, they are essential for transition to the repair process. They secrete important growth factors such as transforming growth factor alpha, transforming growth factor beta, fibroblast growth factor, vascular endothelial growth factor, and also the platelet derived growth factor is released in this environment. The growth factor, basically they have got various function, specific functions such as the VEGF, vascular endothelial growth factor, stimulate new blood vessel formation. The fibroblast growth factor, it causes the proliferation of the fibroblast, and also it is responsible for new matrix production, extracellular matrix production. The platelet-derived growth factor is essential for recruitment of fibroblast and smooth muscle cell in this environment. The transforming growth factor beta, it is important for promotion of fibroblast activation, extracellular matrix production, and scar formation, while transforming growth factor alpha stimulates the epithelial cell proliferation. The fibroblast activation and migration is fibroblast activation and migration to this environment is essential for the healing process. New blood vessels that are formed under the stimulation of VEGF and also by fibroblast growth factor, they are also important, and this epidermal migration is assisted by the transforming growth factor alpha. Overall purpose of this stage is the transition from inflammatory to the repair, and it is marked by clearance of the debris, secretion of the growth factors, and activation of the fibroblast along with angiogenesis and initiation of the granulation tissue formation. This process occurs between the day 2 to 4. Here a later stage of the wound healing is depicted. This is day 4 to day 8. Here this illustration is focusing on granulation tissue formation and degradation of thrombus to allow new tissue development. The key components over here are thrombus. The original fibrin platelet is still visible. The plug platelets and this fibrin plug is still visible, but it is now broken down, and the key enzymes that are involved in this process are urokinase plasminogen activator and tissue plasminogen activator, along with matrix metalloproteinases. These molecules degrade the fibrin mesh and extracellular matrix components, clearing the way for the new tissue. So the granulation tissue is formed below the thrombus. There is proliferation of the fibroblast down there. Over here, there is proliferation of the fibroblast. They are proliferating and forming collagen. New capillaries are sprouting into the wound area, and the dermis now contain fibroblast, blood vessels, and loose collagen fibers. This creates the scaffolding for the permanent tissue repair. The biological process over here are fibrin and temporary matrix that are dissolved by urokinase plasminogen activator and tissue plasminogen activator, that activating plasmin to digest the fibrin, and matrix metalloproteinases that are breaking down the matrix protein. This granulation tissue that is formed, that has started to form over here, it grows upward and fills the gap that is created by the wound. Simultaneously, the migrating epithelium, the epithelium that was under the influence of tissue uh, transforming growth factor, under the influence of transforming growth factor, it edges the wound and eventually it covers the wound. The efficient thrombus clearance is essential for the healing, and poor regulation can lead to chronic non-healing wound if the degradation of this thrombus is insufficient. Excessive breakdown of this, if not accompanied by the granulation tissue formation, may result in weak scar development. So here this flow diagram is showing the event that occurs during the process of healing. If there is any injury, there will be formation of the platelet plug, and then in this environment there is release of the various growth factor. These can be platelet derived growth factor, transforming growth factor beta, fibroblast growth factor, and vascular endothelial growth factor, that will lead to the recruitment of the fibroblast and proliferation. The fibroblast starts secreting collagen 3, and later on this collagen 3 is replaced by collagen 1, and granulation tissue is formed. And then this granulation tissue that was formed initially, it will be transformed or remodeled into the dense fibrous tissue scar due to presence of collagen type 1. Then these growth factor can also lead to mechanical tensions and exposure to transforming, especially the exposure to the transforming growth factor beta, that will lead to the conversion of fibroblast, transformation of the fibroblast into myofibroblast. There will be a smooth muscle actin presence, and the rearrangement of the smooth muscle actin then will lead to the wound contraction. There is a wide variation in the repair site. The amount of local tissue destruction differ by context. In surgical excision, there is or in skin lesion, there is little or no dead tissue, while in myocardial infarct, that is medium size, there is localized demarcated necrosis. In large third degree burn, the necrosis of the tissue is widespread and irregular. Then, if there is injury, especially the epithelial tissue, there such as skin, there is a role of resident gamma delta T cells that are called dendritic epidermal T cells or DETs. So their function is to sense local injury, initiate the cellular immune response, and support the keratinocyte proliferation and survival by releasing some growth factor, chemokines, and cytokines. Initial acute inflammatory response is dominated by neutrophil, and its purpose is to liquefy and clear the necrotic tissue. Acute inflammation persists if necrotic debris remains. Bacterial infection is persistent. So these must be cleared before repair, and after their clearance or their entrapment within the scab, the granulation tissue formation and repair can proceed. Before the granulation tissue appear, the exudative neutrophil can form pus. So there is secretion or formation of fibronectin, matricryptin, that are the matrix-derived signaling fragments, or chemokines or cellular debris. They recruit macrophage and fibroblasts to this area. A fibrin clot is formed during 0 to 4 hours of the injury, and in this diagram it is illustrated uh, along with the early organization of the extracellular matrix and its function in the wound healing. And here it is also focusing on the role of M1 macrophage on the and the fibronectin and activity of the transglutaminase. So M1 macrophage, they are also named as classically activated macrophage, and they are involved in clearing of the dead tissue and pathogen and initiation of the inflammation. At this stage, they also help to organize the provisional matrix by releasing signals. In the center, this fibrin network is also seen, and the wound bed is dominated by fibrin fiber. They are arranged in loose mesh, and this fibrin is providing the initial scaffold that is necessary for cell migration and tissue reconstruction. The fibronectin that is linked to the fibrin, they are attached to the fibrin fiber. Their function is to promote cell adhesion and cell migration, and they serve as guide for fibroblast and endothelial cells. There is also role of transglutaminases, and these transglutaminases such as factor 13 or tissue transglutaminase, they are responsible for cross-linking of the fibrin and fibronectin. Their purpose is to strengthen and stabilize the provisional matrix, and they also support initial mechanical strength of the wound. Early stabilization of the matrix is crucial for the prevention of the wound dehiscence or rupture of the wound that allows subsequent fibroblast and endothelial migration. If this transglutaminase activity is excessive, it can contribute to fibrotic scarring. This diagram is illustrating the importance of the dual role of macrophages in the phagocytosis of the debris and matrix remodeling. The macrophage is a large cell. This macrophage that is depicted in this diagram by this blue area. This macrophage is a large cell, and it has got a large visible nucleus, and it is engaged in both defense as well as the repair role during the healing of the wound. So for the phagocytosis of the pathogen, the phagocytosed bacteria or debris, they are enclosed within a phagosome. This green vesicle is a phagosome, and in this phagosome there is presence of this phagocytosed material that is present inside, and this internal digestion of the pathogen, it prevents infection and clears the wound bed. Then there is also secretion of the proteolytic enzymes, and these proteolytic enzymes that are secreted by the macrophages in the extracellular spaces, they are responsible for degradation of the old matrix protein like fibrin, fibrinectin, and type 1 collagen. So this degradation is essential for clearing the damaged tissue and making space for new matrix deposition. Then for the matrix remodeling, fibronectin and proteoglycans are either broken down, reorganized, or synthesized anew to help rebuild the tissue structure. Type 1 collagen fiber are laid down and rearranged as part of the scar tissue matures. So the biological importance of this activity is that this activity clears the dead tissue and the microbes, and the proteolytic enzymes are responsible for remodeling and reshaping the extracellular matrix to prepare for the tissue regeneration. So the balance between these two activities, the degradation and synthesis, is crucial for proper healing. So this diagram is illustrating the early phase of the wound repair, that is the extracellular matrix deposition, that early provisional matrix deposition that happens between the days 2 to 4. And here there is a macrophage present, and simultaneously a fibroblast is also working. So there is active involvement of the M1 phase or M1 macrophage and fibroblast. So this M1 macrophage actively releases the proteinases that are proteolytic enzyme, and their main function is to break down fibrin and other provisional matrix protein. And as a result, that cellular debris and other microbes and bacteria, they are also cleared away, and they prepare this area, the wound bed, for the new tissue formation. So proteinases digest the fibrin meshwork and damage fibronectin. These proteinases, they are responsible for the fibrin meshwork digestion and damaged fibronectin, and this step is crucial for the remodeling and making space for the newly incoming matrix. So matrix synthesis is done by this fibroblast. This fibroblast is synthesizing a new matrix components such as the fibronectin that is essential for the cell adhesion and assembly of the matrix protein, and the proteoglycan that maintain hydration and structural support, and type 3 collagen is the early collagen type that is laid down in the granulation tissue and provide initial mechanical strength before being replaced later by stronger type 1 collagen. And the biological importance of this step is that the balance between this matrix degradation and new matrix deposition by fibroblast is very important. It determines the successful wound healing and minimize the scar formation. And if there is imbalance between these two processes, that can cause either chronic wound or excessive fibrosis. So here this illustration is showing the matrix deposition and maturation phase of the wound healing. This is happening between day 2 to day 5. There is gradual fibrin removal and fibroblast producing different materials. So there is active role of the fibroblast in this stage, and the fibroblast, they actively synthesize and secrete collagen type 1 and type 3 both. These blue fibers are these blue one, they are type 3, while these pink fibers are the type 1 collagen. So these both types of the collagen are produced by the fibroblasts. They also produce proteoglycans and fibronectin. They by the production of these material of the extracellular matrix, it is evident that they are main drivers of the new extracellular material construction. At this stage, the collagen type 3, initially produced to form a soft flexible matrix, provides early mechanical support, while collagen type 1, it gradually replaces type 3 collagen and it provides the strong support to the tissue. Fibronectin is this fibronectin is critical for the matrix reorganization and attachment of the cell. The proteoglycans that are also produced by these fibroblasts, these proteoglycans maintain matrix hydration, provide resilience and binding site for growth factor. So the resident fibrin of this area, this resident fibrin is gradually replaced by the more structured collagen network. So to summarize, we can say that early matrix is composed of loose fibrin and fibronectin network. Mid-stage matrix is composed of type 3 collagen predominantly, and later type 1 collagen predominates and strengthens the scar. The biological importance or significance of this stage is that this organized extracellular matrix formation is essential for restoring the tissue integrity and strength. Also, the proper collagen remodeling ensures the minimal scarring and functional recovery. Disruption in this process can result in weak scar, excessive fibrosis, or chronic wound. This illustration is showing the maturation phase of the wound healing. That is the final phase of repair reaction that starts from the day 5 and takes about weeks to complete. In this phase, the M2 macrophages and fibroblasts, the M2 or alternate macrophages and fibroblasts, they collaborate together and they now continue the process. So they remodel and strengthen the newly formed tissue, And these M2 macrophages or alternately activated macrophages, they are dominantly present in this phase. Their role is to suppress the inflammation, promote the tissue remodeling, and stimulate the activity of the fibroblast. Unlike M1 macrophage or classical macrophages, M2 macrophages support healing and scar formation rather than fighting the infection. So the hybrid collagen fibers are visible in this phase, the final phase. This hybrid collagen fiber type composed of type 1 and type 3, they are visible and they provide strong tensile strength to the tissue. Type 1 and type 3 are flexible but now partially replaced or mixed by type 1 for final tissue strength. The hybrid fibers show the transition toward mature scar tissue. Proteoglycans and fibronectin are also present in this phase, and they are trying to organize the collagen fibrils. The fibroblast activity is still there and they continue to produce and align the collagen fibers. Ribosomes and endoplasmic reticulum inside, they indicate active protein synthesis. Fibroblasts help bundle collagen fibers to enhance the mechanical stability. The biological significance at this stage is that the scar tissue form as collagen fiber thicken and organize. M2 macrophages ensure the resolution of the inflammation and promotion of mature matrix deposition. A successful balance between the collagen type, organized deposition lead to a strong functional scar. So this is the timeline for the collagen dominant scar formation. First, there is uh, formation of fibrin-rich clot with the thrombus formation, and then there is a recruitment of neutrophil and macrophages. Then various growth factors come to this area by the cells. These growth factors are platelet-derived growth factor, transforming growth factor beta, and fibroblast growth factor. And then fibroblasts migrate to the site of the wound. Granulation tissue is formed. Matrix remodeling occurs by the action of matrix metalloproteinases and plasminogen activator tissue and urokinase both. Collagen 3 is deposited first, that is temporary one, then fibroblasts are converted into myofibroblast and replacement of the type 3 by the type 1 collagen occurs and finally a stable scar is formed. Here the role of macrophages is summarized. The macrophages are recruited to the site of the injury. They are bone marrow derived, they can be tissue resident macrophages or bone marrow derived macrophages. They first are responsible for inflammatory reactions, and then after the release of various proteases that degrade the extracellular matrix and engulfment of phagocytosis of the debris and the pathogen, they also convert into alternate macrophage, and they are now M2 M1 type are not there. M2 type of the macrophages, they are responsible for resolution and of the inflammation. There is decreased fibroblast activation and collagen, that is type 3 is replaced by type 1 and a scar is formed, and these alternate macrophages are responsible for decreasing the tissue damage and infections. So alternate type of the macrophages are important for healing, while the classical macrophages are important for the inflammatory phase. So here a detailed flow chart of macrophage function and transition during the tissue injury and healing is described. So the macrophages are sequestered in the spleen, or they come from bone marrow as monocyte, and these monocytes are they reach to the site of the inflammation or site of the injury by the blood, and here uh, the polymorphonuclear neutrophils also join them, and the granules of these neutrophils, they are responsible for the recruitment of the monocyte, further recruitment of the monocyte to this area, and at the site they are transformed either into monocyte-derived tissue inflammatory phagocytic dendritic cells or inflammatory macrophages. So, further, there is activation of T helper cells. So T helper 1, T helper 2, and Th17 responses of the inflammation are also activated, and Langerhans cell activation and dendritic cell uh, involvement lead to the further neutrophilic recruitment that accounts for the immune response that is associated with it. So there is also proliferation of the macrophages that are resident to that area, and they are more in number now in this area. Then the classical activation or M1 macrophage activation is by the lipopolysaccharide or interferon gamma, and these M1 or classical macrophage form pro-inflammatory cytokines, phagocytosis, and they also are responsible for fibrin and matrix removal via matrix metalloproteases. They also release factors such as VEGF, platelet-derived growth factor, interleukin-1, interleukin-6, and tumor necrotic factor, with many chemokines. So there is a spectrum of the macrophage plasticity. They can shift from pro-inflammatory M1 to the healing M2 based on the environment of the cytokines. And there is also phagocytosis. These phagocytes are responsible for phagocytosis, and this phagocytosis is responsible for the clearance of the apoptotic neutrophils and other material that is present in that environment, and lead that environment towards the healing environment. Then the alternatively activated macrophages that are driven by interleukin 4, interleukin 13, they are formed. They are also named as M2 macrophages, and their function is anti-inflammatory and activation of the fibroblast, along with angiogenesis and tissue remodeling. Then the fibroblast proliferation and activation is due to release by the macrophages of transforming growth factor beta, platelet-derived growth factor, and interleukin 10. And they promote not only the proliferation but also the transformation of the fibroblast into myofibroblast, and then collagen is deposited and a scar is formed. Regulatory macrophages and the T cells have got also some role. The regulatory macrophages and T regulatory cells finalize the healing and suppress excessive inflammation. So the main phases are inflammation, that is M1 dominant. Then there is transition, and then there is healing macrophage, that is M2 macrophage. So the repair process begins when macrophage uh, predominate at the site of the injury. The inflammatory type or inflammatory functions are due to classical macrophages, while the alternate type of the macrophages are basically meant for uh, meant to drive the tissue towards the healing process. Thank you for watching this video. If you have any query, please write in the comment box.